Deep within the wet prairies of the Pacific Northwest, there is this uh, prized, sugar-rich food staple. But it grows so agonizingly close to a lethal, nerve-paralyzing look-alike that literally a single, careless shovel stroke can unearth both life and death.
Yeah, it really is the ultimate high-stakes harvest. I mean, you are quite literally pulling your survival or your demise from the exact same patch of soil. The margin for error is just absolute zero.
Right. So for you listening right now, imagine putting on your ethnobotanist hat with us today,
Yeah.
because we're going to unpack a story of, well, profound botanical complexity, ancient survival, and absolute thermodynamic mastery.
It's a fascinating landscape to look at.
It really is. Our mission today is to explore that razor-thin biological line between the edible blue camas, known scientifically as Camassia quamash, and the incredibly toxic meadow death camas, Toxicoscordium venenosum.
Exactly, and we're also going to dissect the molecular chemistry of those hazards. We'll look at the specific neurotoxic mechanisms that make mistakes so unforgiving, and then we'll break down the ingenious, really just brilliant thermodynamic engineering that indigenous peoples developed over millennia to safely turn an otherwise completely indigestible root into the cornerstone of a civilization's diet.
But before we get into the roots and the biochemistry, there is a crucial disclaimer we have to state up front. While we are going to explore the botanical features of these plants in extreme detail today, this deep dive provides zero foraging instructions.
Zero. None at all.
Right. The lethal consequences of this identification are far too severe to risk. We are analyzing this as a marvel of ancient science and ecology, not a how-to guide for your next hiking trip. So please do not go out and eat these plants based on what we discussed today.
Yeah, I mean, the toxicology reports we're going to cover a bit later should be more than enough to dissuade anyone from taking that gamble.
Absolutely. So to really understand how this evolutionary drama plays out, you have to visualize the ecological theater where these two plants coexist. We are talking about the moist meadows, the vernal wetlands, and these sun-drenched lowland soils of western North America.
Right. If you picture the Pacific Northwest stretching from southern British Columbia down through Washington, Oregon, and into California, and even eastward toward Montana and Wyoming, that is prime Camas country.
But there is a very specific archipelago within the Salish Sea, right? The San Juan and Gulf Islands. And it sits in the rain shadow of the Olympic Mountains, which creates this totally fascinating microclimate.
The rain shadow effect is critical here, actually. Because the Olympic Mountains block the major storm systems coming off the Pacific, this specific archipelago only receives about half the precipitation of a place like, say, Seattle.
Oh, wow. I didn't realize the drop-off was that severe.
Oh, it's massive. So you end up with these incredibly diverse, compressed ecological zones. Like you've got native prickly pear cacti growing on rocky, sun-baked bluffs, literally right next to windy sand dunes.
That sounds like a completely alien landscape for the Pacific Northwest.
Right. And nestled right within these contrasting zones are the wet, vernal prairies. These are areas that flood in the early spring, creating temporary wetlands, and then they bake completely rock-hard dry by late summer. It's a harsh, really demanding ecological cycle.
And it's right in those wet early spring soils that our two main characters emerge side by side, the edible blue camas and the toxic death camas. And for anyone walking through those fields, the terrifying reality is that they are morphological mimics for most of the year.
They really are. If you look at them during their vegetative phase, the similarity isn't just close, it's practically identical.
Yeah, they both emerge early in the spring, setting up these narrow, V-shaped, grass-like leaves from an underground bulb.
And the bulbs themselves share the exact same architecture. Botanists refer to them as tunicate bulbs.
Tunicate like a tunic?
Exactly, like a layered garment. A tunicate bulb is constructed of concentric, fleshy scales protected by a dry, papery outer coat. When you pull them out of the earth, both the blue camas and the death camas look remarkably like a small wild onion or a shallot.
They have that same dark, flaky exterior and the same layered interior.
They do. But there is a massive missing variable that often traps the unwary forager. Neither of these plants produces an onion-like scent.
Which is wild because they look exactly like onions.
Right, but they completely lack the volatile sulfur compounds that give plants in the Allium genus their characteristic smell.
And that absence of scent is a massive liability for modern foragers, isn't it?
It's the #1 reason people get poisoned. People hiking through these prairies will spot the bulb, note its tunicate structure, and just assume it's a wild onion.
But they don't smell anything.
Right. When they don't smell that sharp allium odor, they rationalize it. They might assume the plant is just too young or, maybe it's growing in the wrong soil to produce the scent. And if the bulb they happen to have dug up is Toxicoscordium venenosum, the death camas, that rationalization is fatal.
I mean, there are sobering medical case studies documenting this exact mistake. While reading through our sources for this deep dive, I found a recorded case from 1994 involving a 50-year-old Navajo man.
Oh, I know the case you're talking about.
Yeah, it's terrifying. He was out hiking with his daughters and dug up what he confidently identified as wild onions. And he just ate them raw right there.
Raw death camas.
Yes. Within a single hour, his body began shutting down completely. He experienced severe neurological symptoms, profuse vomiting, and a massive, highly dangerous drop in blood pressure.
He was incredibly lucky to receive rapid emergency medical care and actually survive that.
He really was. The hospital later analyzed the remaining plants he'd collected in his bag and confirmed they were Toxicoscordian.
Which just highlights the immense danger of foraging in these regions without deep generational knowledge, because there is only one brief period in the entire annual cycle of these plants when you can safely differentiate them using just your eyes.
Right, the narrow window of clarity.
Yeah.
It happens during their blooming period, which usually runs from late April through mid-May, depending on how high up in elevation you are.
Yeah, elevation changes the timing slightly, but it's a short window.
So if you are standing in a prairie in early May, the visual distinction finally reveals itself. The edible blue camas, Camassia quamash, sends up a tall, striking inflorescence. It forms what botanists call the simple terminal raceme.
Right, and to visualize a terminal raceme for our listeners, just picture a central, unbranched stalk shooting straight up. And spaced along this central stalk are individual flowers, each attached by its own short little stem, sort of like grapes hanging off a main vine.
And the flowers on the blue camas are genuinely breathtaking. They're showy, quite large, with 6 distinct petals ranging in color from a pale icy sky blue to a deep, vibrant violet purple.
They're gorgeous.
Yeah, when Meriwether Lewis of the Lewis and Clark expedition first saw these fields in June of 1806, he wrote in his journal that from a short distance, the blue camas fields looked exactly like lakes of fine, clear water.
That's a perfect description. You can imagine thousands of these blue racemes swaying in the wind, mimicking the surface of a lake.
But hidden within that beautiful blue lake is the assassin.
The meadow death cam.
Exactly. It also blooms in that same window. But its inflorescence is structurally different. It forms an unbranched raceme or occasionally a looser branching structure called a panicle. And the flowers themselves are much smaller.
Yeah, instead of large blue petals, the death camas produces tightly clustered, creamy white or slightly greenish white flowers.
And if you were to get incredibly close, which, again, you should never do with the intent to harvest, you would notice a specific structural difference in the flower itself. The death camas has tepals.
Tepals are such a fascinating botanical feature. Usually a flower has green sepals underneath to protect the bud and colorful petals on top to attract pollinators. But in some plants, the sepals and petals look identical, blurring into a single structure.
Like a tulip.
Exactly like a tulip. Those are tepals. The death camas has 6 of these creamy white tepals, and right at the base of each one, there is a distinct, thickened, yellow-green nectary gland.
It looks almost like a little droplet of yellowish wax just baked right into the flower.
That's a great way to describe it. And the edible blue camas does not have these glands.
Okay, so you have a few weeks in the spring where the white flowers and the blue flowers announce themselves. But once mid-May passes, those flowers fade, they dry up, and they drop their seeds. The stalks wither away entirely.
Back to square one.
Right. By late summer, which is the traditional time to actually harvest the bulbs for maximum carbohydrate yield, you are back to looking at identical dried grass-like leaves and identical tunicate underground bulbs. I'd had to challenge the sheer survival logic here.
It does seem crazy to a modern observer.
It really does. Harvesting dormant camas late in the season sounds like playing botanical Russian roulette in the dark. If they look identical underground, why would human beings ever risk digging them up?
Well, the indigenous agricultural methods made it safe, which we'll get into.
Right. But before we explore that incredible methodology, we need to understand the stakes. What happens if you lose that game of Russian roulette? Let's talk about the chemistry of the poison.
Okay, let's get into it. The lethality of the death camas stems from a highly complex steroidal alkaloid neurotoxin called zygosine. And what makes Toxicoscordium venenosum so relentlessly dangerous is the distribution of this toxin.
Because some toxic plants only concentrate their defenses in their roots or maybe their seeds, right?
Exactly. The death camas is different. It saturates its entire physiological structure with zygosine. The fibrous leaves, the papery outer coat of the bulb, the dense inner layers, the stem, the petals, even the nectar and the pollen are heavily laced with this neurotoxin.
So touching any part of it, or worse, eating any part of it, is a disaster.
A complete disaster.
From what I understand, the zygosine acts as a chemical wedge that attacks the human nervous system at the most foundational microscopic level.
It really does. In every nerve and muscle cell in your body, you have these tiny pores embedded in the cell membrane called voltage-gated sodium channels. They are classified scientifically as Navi 1.1 through Navi 1.9.
I try to think of these channels as microscopic floodgates, holding back a reservoir of sodium ions.
That's a good analogy. When your brain wants to send a signal, say, to move your arm or even just to keep your heart beating, it sends an electrical impulse called an action potential. When that electrical charge hits the cell membrane, those sodium floodgates instantly swing open.
And the sodium rushes in.
Sodium ions rush into the cell, carrying the electrical signal forward, kind of like a stadium wave moving through a crowd. But for your nervous system to function properly, that wave has to pass quickly, and the gates must immediately close to reset the system for the next signal.
And the mechanism that shuts those gates is a tiny molecular hinge called the IFM motif, right?
Yes, it stands for isoleucine, phenylalanine, and methionine. It's basically a hydrophobic, water-repelling plug that swings into the channel and snaps the floodgate shut within a fraction of a millisecond.
The signal passes, the gate closes, your body remains stable.
Precisely. But when zygosine enters the bloodstream, it seeks out those specific sodium channels. The zygosine molecule is physically shaped in such a way that it binds to the channel and wedges itself right into the pathway of that IFM inactivation gate.
Oh, wow. So it physically blocks it from closing.
It acts like a boot stuck in a doorway. The gate tries to swing shut, but the zygosine blocks it. The sodium channels become permanently locked in the open position.
So the floodgates are just jammed open. Your cells are completely inundated with a massive, uncontrolled influx of sodium and calcium ions.
The cellular environment is completely overwhelmed. This continuous, unchecked electrical stimulation forces the nerves to rapidly deplete their neurotransmitters. Specifically, it triggers a massive dumping of acetylcholine into the synapses.
And acetylcholine is the primary neurotransmitter that controls the autonomic nervous system, right? The system that runs all the involuntary functions of your body.
Exactly. Digestion, heart rate, glandular secretions, all of it.
So when your entire body is suddenly drowning in acetylcholine, the result is a systemic neurological short circuit.
It's chaos. Toxicologists categorize this specific type of poisoning under an acronym known as the SLUDGE toxidrome. And it is as horrifying as it sounds.
Walk us through the acronym because it sounds awful.
SLUDGE stands for salivation, lacrimation, which is uncontrollable, tearing of the eyes, urination, defecation, gastrointestinal upset, and emesis, which is severe vomiting.
Your body essentially begins violently purging fluids from every single orifice because the autonomic nervous system has lost all ability to regulate itself.
It's just voiding everything. And the terrifying part is that the SLUGE symptoms are merely the first stage of the crisis.
It gets worse.
Much worse. As the zygosine binds to more and more sodium channels throughout the body, the cardiovascular system begins to collapse. Blood pressure plummets to dangerously low levels.
And you lose motor control, too.
Right. The victim experiences ataxia, which is a complete deterioration of voluntary muscle coordination. You can't walk. You can't balance. There's this terrifying tingling and burning numbness that starts in the mouth and throat and rapidly spreads out to the fingers and toes.
I read that as the respiratory muscles lose their ability to contract properly, breathing becomes incredibly shallow and erratic.
It does. And eventually, this culminates in severe convulsions, a deep coma, and fatal respiratory or cardiac failure.
The sheer potency required to trigger this entire cascade is terrifyingly small. I mean, as few as 1 or 2 mature death Camas bulbs contain a sufficient concentration of zygosine to deliver a fatal dose to a fully grown human being.
1 or 2 bulbs. Think about how easy that is to accidentally ingest if you think you're eating wild onions.
And you cannot cook the danger away, right?
No, absolutely not. Zygosine is an incredibly robust, heat-stable molecule. Boiling the bulbs, roasting them, drying them out, none of it degrades the toxin. Researchers have actually tested dried death Camas bulbs that sat on a herbarium shelf for 20 years, and they were still lethally toxic.
That is wild. In laboratory tests, the oral LD50, which is the median lethal dose required to kill 50% of a test population of zygosine in mice, is shockingly low. It's just 132 milligrams per kilogram of body weight.
To put that in perspective for agriculture, if a cow or a sheep happens to graze on just half a pound of the green leaves emerging in the spring, it is enough to kill the animal outright.
Just half a pound.
Yeah, it causes devastating losses for modern ranchers who let their livestock graze in these unmanaged wet prairies. The animals lose coordination, start foaming at the mouth, and just drop dead in the fields.
While preparing the research for this deep dive, I came across a really harrowing first-hand account on a wilderness medical blog.
Oh, is this the story about the boy in the mountains?
Yes. A man named Troy was recounting a survival trip he took into the mountains when he was just 12 years old. He was out foraging and mistakenly identified a cluster of bulbs. Over the course of the afternoon, he ate 12 deaf camas bulbs. 12 of them.
That is a miraculously lethal ingestion. I mean, the fact that he lived to write about it defies all pharmacological odds.
His survival was an absolute agonizing nightmare. He had to be emergency evacuated and rushed home. By the time he reached a medical facility, the doctor explicitly told his mother there was virtually nothing they could do.
Because the zygosine had already locked the channels open.
Right. It had fully saturated his nervous system, and too much time had elapsed for stomach pumping to be effective. His heart rate plummeted to 20 beats per minute.
20 beats per minute is barely alive.
He described lying in the hospital bed, completely paralyzed, having to consciously, physically force his body to take every single breath, because his autonomic nervous system had simply quit firing the signals to his diaphragm.
He was experiencing the final stages of respiratory failure, kept alive only by his own desperate conscious will to inhale. It's horrifying.
He somehow endured the chemical storm and survived, but the neurological damage was profound. He wrote that he suffered from severe, debilitating migraines and lingering respiratory issues for years afterward.
That is the power of the chemical weapon we are dealing with.
But looking at this from an evolutionary standpoint, I have to push back on the plant's biological strategy here. It seems completely counterproductive.
How so?
Well, a plant expends massive amounts of metabolic energy to grow a tall stem, unfurl showy flowers, and produce nectar, right? The entire biological purpose of nectar is to bribe insects into visiting the flower, getting covered in pollen, and flying away to fertilize another plant.
Right.
So why would the death camas lace its own nectar and pollen with a fatal neurotoxin? You are effectively murdering your own delivery drivers. It looks like evolutionary suicide.
It really appears that way until you observe the specific insect ecology surrounding the plant. The death camas has evolved a highly exclusive, almost symbiotic relationship with a solitary ground-nesting insect called the death camas minor bee, scientifically known as Andrena ostragali.
Wait, so there is a specific bee that has cracked the chemical code of zygosine?
Yes. The death camas minor bee is what entomologists call an oligolage.
An oligolage.
Yeah, an oligolage is a specialized pollinator that exhibits a narrow, highly restricted dietary preference. They often forage for pollen from only a single genus or even a single species of plant. And Andrena ostragali is likely the only insect in that entire Pacific Northwest ecosystem capable of tolerating zygosine.
That is incredible. So by poisoning its own nectar, the death camas is building an evolutionary moat around itself.
That is the brilliant, ruthless logic of it. Think about the competition for pollinators in a spring meadow. You have hundreds of plant species blooming simultaneously, all desperately trying to attract honeybees, bumblebees, and butterflies.
It's a crowded market.
Exactly. The death camas bypasses that competition entirely. By lacing its nectar with a neurotoxin, it effectively assassinates any generalist pollinator that attempts to steal its resources. It just kills the competition.
And researchers actually see the evidence of this in the field, right? If you find a death camas plant in bloom, you will frequently find a literal graveyard at its base.
Rings of dead or paralyzed flies, beetles, and non-adapted bees lying in the dirt.
They landed, took a sip of the sweet nectar, and the zygosine locked their sodium channels open before they could even launch themselves back into the air.
It's a lethal VIP banquet. But for the minor bee, this highly toxic environment is a sanctuary. Because no other insect can survive the nectar, the minor bee has a guaranteed, uncontested food source.
And the evolutionary benefits likely flow both ways, don't they?
Oh, definitely. The female minor bee collects this highly toxic pollen and nectar, carries it back to her underground nest, and forms it into a dense provision mass to feed her larvae. By rearing her young on a diet of neurotoxins, she is potentially giving them a chemical shield, making the larva highly unpalatable or outright toxic to predators and soil parasites.
A perfectly closed, co-evolved loop of toxicity. But humans don't have that evolutionary cheat code. We are generalists. We don't have a biological shield against zygosine, which makes the fact that human beings chose to eat the plant sitting right next to it completely baffling.
It is quite the bold culinary choice.
So imagine you are a native inhabitant of this region thousands of years ago. You learn, through tragic trial and error, to avoid the white flowers. You successfully navigate the minefield. You dig up a basket full of beautiful, safe blue Camas bulbs. You wash the dirt off. You have finally secured a nutritious meal, right?
You have secured a botanical specimen. You have absolutely not secured a meal.
Wait, but after avoiding the poison, after pulling the right bulb out of the ground, I still can't eat it.
If you bite into a raw Camassia quamash bulb, you're going to experience a completely different, though non-lethal form of physiological misery. Raw blue Camas is entirely indigestible to the human gastrointestinal tract.
Okay, let's break this down. Because if you look at a raw Camas bulb, it looks remarkably like a small, dense potato. And potatoes are the ultimate survival food. They are packed with energy. Why does the human body treat a Camas bulb differently than a potato?
The answer lies in the microscopic architecture of the carbohydrates. During the summer, plants photosynthesize sunlight into simple sugars. To survive the dormant winter and have the energy to sprout new leaves in the spring, they have to pack those sugars away to storage molecules.
Like starch.
Exactly. Potatoes, like many common root vegetables, store their energy as starches. Starch molecules are composed of long chains of glucose linked together by what chemists call alpha-glycosidic bonds.
And human biology is perfectly adapted to deal with alpha bonds.
We are perfectly adapted. Human saliva and the pancreatic juices secreted into our small intestines are loaded with an enzyme called amylase.
I think of amylase as a highly specific chemical key designed to unlock one specific lock.
That's spot on. When you chew a piece of raw or cooked potato, the amylase instantly binds to those alpha-glucan chains and rapidly breaks the bonds, shearing the massive starch molecule into hundreds of individual simple glucose molecules.
Your intestines absorb the glucose,
Yeah.
and you get a massive spike of instant usable energy.
But the blue camas did not evolve to use starch. It belongs to a family of plants that stores its winter energy reserves as a completely different complex carbohydrate called inulin.
Inulin. If you look at the nutrition label on modern protein bars, keto snacks, or prebiotic supplements, you'll often see inulin listed as an ingredient.
You will, and it is usually derived from chicory root or agave. Inulin is a type of fructose. Chemically, it is a massive structural polymer composed almost entirely of fructose molecules.
So, unlike glucose in potatoes, it's fructose.
Right. Imagine a heavy anchor chain made of 30 to 60 individual fructose links. And the critical difference is how those links are welded together. Instead of the alpha bonds found in starch, the fructose molecules in inulin are fused together by beta 2-to-1 glycosidic bonds.
Beta 2-to-1 bonds.
Yes. Often there is a single glucose molecule capping the very end of this massive chain, but the bulk of the structure is held together by these beta 2-to-1 linkages.
And I'm guessing that our amylase enzyme, our chemical key that works so well in potatoes, does absolutely nothing to a beta bond.
Human digestive enzymes are completely blind to a beta 2-to-1 glycosidic bond. The physical shape of the inulin molecule prevents our enzymes from gripping it.
So it's the wrong key for the lock.
Completely the wrong key.
Yeah.
So when a human being chews and swallows a raw or even a conventionally boiled blue Camas bulb, those massive 60-link inulin chains pass completely untouched through the highly acidic environment of the stomach.
They just sail right through.
They travel right through the entire length of the small intestine without a single calorie being extracted.
From a nutritional standpoint, eating raw Camas is like eating sawdust or a handful of cotton balls. The energy is there, but your body lacks the chemical tools to access it. So it just acts as rough dietary fiber and passes straight through you.
Which is pretty grim if you're starving.
Right. But that doesn't sound like a crisis, just a very disappointing meal.
If the inulin merely passed through silently, it would just be disappointing. But the crisis begins when those massive, fully intact inulin chains finally drop into the large intestine, the colon.
The microbiome.
Exactly. Your colon is an active ecosystem housing tens of billions of gut bacteria, and while your mammalian enzymes cannot break down a beta 2-to-1 bond, the bacteria in your lower gut absolutely thrive on them.
To them, a sudden influx of inulin is an all-you-can-eat buffet.
It's a feast. They immediately swarm the carbohydrate chains and begin rapidly fermenting them.
And bacterial fermentation in an enclosed biological space produces gas.
Massive, incapacitating volumes of gas. As the bacteria rapidly metabolize the fructose chains, they generate huge quantities of carbon dioxide, hydrogen, and methane gases as metabolic byproducts, along with short-chain fatty acids.
Oh, man.
Because a meal of raw or poorly prepared camas introduces such a staggering volume of complex fructans all at once, the resulting bacterial fermentation is practically explosive.
We have to put the listener in the shoes of someone experiencing this. The sheer volume of gas causes extreme abdominal distension. Your intestines are swelling like a balloon trapped inside your abdominal cavity.
And it gets worse chemically, too.
Because these undigested carbohydrates are highly osmotic, right? They draw water out of your body's tissues and flood it into the bowel.
Yes. This combination of trapped gas and sudden water influx leads to agonizing, doubled-over cramping and severe osmotic diarrhea. It isn't just uncomfortable, it is a profoundly incapacitating physiological event.
There are famous historical accounts that perfectly illustrate the misery of this specific biochemical reaction.
The Lewis and Clark story is probably the most famous.
Yes. The Lewis and Clark expedition provides one of the most vivid and, frankly, agonizing examples. It's September of 1805. The Corps of Discovery has just spent weeks desperately trying to navigate the freezing, brutal terrain of the Bitterroot Mountains.
They were in terrible shape.
They are lost, they are freezing, and they are literally starving to death. They have been reduced to eating their own pack horses and chewing on candle wax. They finally descend out of the mountains onto the Whitey Prairie in present-day Idaho, and they encounter the Nez Perce people.
The Nez Perce, seeing these starving strangers, graciously offer them the staples of their diet.
Right. They offer them dried salmon, berries, and large baskets of camas bulbs. Now, the Nez Perce knew how to properly prepare camas, but the explorers, driven mad by starvation, simply gorged themselves.
They probably ate much of it raw or just hastily roasted over an open fire.
Exactly, without the proper multi-day preparation. The journal entries from William Clark over the next few days are grimly hilarious in hindsight, but must have been absolute torture to live through.
I can only imagine.
He documents the entire expeditionary force being brought to its knees. Grown men who just crossed the Rocky Mountains were lying on the ground, completely immobilized by severe gastrointestinal distress and explosive flatulence. Clark wrote that the men were complaining of a lax and heaviness at the stomach.
David Douglas, the famous Scottish botanist for whom the Douglas fir is named, explored the Columbia River region a couple of decades later in the 1820s. And he had a very similar, poorly timed encounter with the root.
What happened to him?
He wrote in his botanical journal about staying overnight in a small, enclosed indigenous lodge. He partook in a large meal of camas and dryly noted in his journal that they produce flatulence. And when in the Indian hut, he was, quote, blown out by strength of wind.
Blown out by strength of wind is an incredible historical visual. A renowned Scottish botanist forced to flee a hut in the middle of the night because of weaponized inulin fermentation.
It's quite the legacy.
So the raw blue camas won't kill you like the zygosine and the death camas, but it will absolutely make you wish you were dead. This brings us to the most spectacular part of the deep dive. How did indigenous peoples crack this chemical code?
This is where it gets truly amazing.
How do you take a rock-hard bulb of completely indigestible inulin and transform it into a calorie-dense, sweet staple food without turning your entire village into a balloon factory?
They had to devise a method to systematically break those stubborn beta 2-to-1 bonds before the food ever entered the human digestive tract. And what is truly astonishing is that they didn't just stumble upon a basic cooking technique. They essentially engineered an underground acid-catalyzed chemical reactor.
I'm going to walk through the exact blueprints of this reactor, because my first instinct, and probably the instinct of anyone listening, would be to just take the bulb and throw it in an oven or roast it in the coals of a fire. If I want to cook a potato, I bake it. Why does simple dry baking fail to unlock the calories in camas?
If you take a camas bulb and put it in a modern dry oven, setting the temperature anywhere between 135 and 190 degrees Celsius, which is standard baking temperature, the inulin does not break down into simple digestible fructose sugars.
It just stays intact.
Worse, dry heat without the presence of trapped moisture, and crucially without an acid catalyst, causes the massive inulin chains to degrade improperly. Instead of breaking apart into individual usable links, the heat forces the chains to twist and fuse into complex cyclic compounds called didifructose dianhydrates, or DFDAs.
So the chemical structure just scrambles itself into rings. And let me guess, human digestive enzymes are completely blind to DFDAs as well.
Exactly right. DFDAs are utterly indigestible. You might soften the physical texture of the bulb in the oven, making it easier to chew, but you have fundamentally failed to solve the chemical puzzle. You will extract 0 calories from it, and you will still suffer the gastrointestinal consequences.
So baking is a bust.
Total bust. To correctly cleave those beta bonds and yield bioavailable calories, you need 3 precise variables occurring simultaneously: sustained, moderate heat, high moisture, and a significantly lowered pH. You need an acidic environment.
So how did the Coast Salish, the Nez Perce, the Kalapuya, and other indigenous groups across the Pacific Northwest achieve this highly specific biochemical threshold thousands of years before the invention of the modern laboratory?
Through the precise thermodynamic engineering of the traditional earth oven, commonly referred to as pit roasting. The construction of these ovens was a massive communal undertaking that required deep empirical knowledge of physics and botany.
How they build it.
First, they would dig a large, deep, bowl-shaped pit into the earth, sometimes several feet across and several feet deep. Inside this pit, they would build a massive roaring fire. Once the fire was established, they would load it with a bed of high-density rocks.
Just any rocks.
No, they specifically selected basalt or river rocks that had been carefully vetted, because the wrong type of rock could contain trapped moisture and literally explode like shrapnel when superheated. They would heat these rocks until they were glowing red hot.
Okay, so you have a subterranean pit filled with superheated thermal mass. What is the next step?
The remaining embers and ash are quickly cleared away or tamped down, and those superheated rocks are immediately covered with thick, densely packed layers of specific wet native vegetation.
And they didn't just use whatever leaves were lying around, right?
Right. They specifically gathered skunk cabbage leaves, the green boughs of western red cedar, sword ferns, and sometimes medicinal plants like kinnikinick or salal.
Skunk cabbage is an incredibly pungent, foul-smelling plant. It literally smells like a skunk. That doesn't sound like something you want anywhere near your food.
They weren't using it for culinary flavor. They were using it for its specific chemical properties. Once the thick layer of wet vegetation was laid over the glowing rocks, the raw blue Camas bulbs, sometimes hundreds of pounds of them, gathered by the entire community, were piled directly on top.
Wow, hundreds of pounds at once.
Yeah, it was a massive harvest. Then, more thick layers of wet vegetation were placed over the bulbs, encasing them completely. Finally, the entire pit was rapidly sealed shut with earth, packing the dirt down tightly to ensure that not a single wisp of steam could escape.
You have essentially buried a giant pressure cooker.
A pressure cooker with a massive thermal battery, and they would leave it sealed beneath the earth for an exceptionally long time. Depending on the size of the harvest and the moisture content of the bulbs, the oven would remain closed for anywhere from 24 to a staggering 72 hours.
Up to 3 full days underground. That is an immense investment of time and resources. What exactly is happening chemically in the dark during those 72 hours?
This is where the brilliant intersection of thermodynamics and chemistry occurs. Deep underground, the glowing basalt rocks begin to vaporize the moisture held within the wet skunk cabbage and cedar boughs. But it is not just pure water vapor rising through the pile.
Let me guess, the skunk cabbage comes into play here.
Yes. As these specific native plants heat up and break down, they release volatile organic acids directly into the steam.
The steam itself becomes acidic.
Precisely. These volatile acids permeate the entire sealed chamber, soaking into the Camas bulbs and drastically dropping the pH of the internal environment. The pH inside the oven plunges to 4 or even below.
Why is a pH of 4 the magic threshold for Camas?
Because the inulin polymer is incredibly stable in neutral or alkaline conditions. You could boil a Camas bulb in a pot of neutral pH water on your stove for 12 hours, and the vast majority of those beta bonds will remain stubbornly intact.
But when you drop the pH to 4,
you fundamentally alter the chemistry. The highly acidic environment significantly lowers the activation energy required to break the bonds. The acidic steam acts as a chemical catalyst.
and combined with the heat.
Combined with the steady sub-boiling temperature maintained inside the sealed pit, which hovers right around 100 degrees Celsius, or 212 Fahrenheit, those massive 60-link inulin chains undergo a process called acid-catalyzed hydrolysis.
Let's visualize that process. Hydrolysis literally means water splitting. Under the intense pressure, the heat, and the acidic conditions, the water molecules are forced into the chemical structure of the inulin. They act like microscopic crowbars, systematically snapping the beta-2-to-1 bonds.
Shearing the massive indigestible chain into thousands of individual free D-fructose molecules.
Fructose, simple fruit sugar, a monosaccharide that the human body absorbs rapidly and effortlessly.
But the chemical transformation doesn't stop at hydrolysis.
There's more.
Oh, yeah. Over the course of the 2 or 3 days, as the inulin is successfully converted into free fructose, the sustained heat radiating from the rocks, which peaks around 110 degrees Celsius, triggers a massive secondary reaction, non-enzymatic caramelization.
Because fructose caramelizes differently than other sugars.
Exactly. Fructose has a uniquely low caramelization point compared to other sugars. As it bakes in that subterranean heat, the complex sugars break down further, forming rich, dark flavor compounds. The hard, white, starchy bulbs slowly transform, turning a deep, glossy, jet black color.
When the earth oven is finally unsealed after 3 days, the transformation must be absolute.
It is. The bulbs emerge incredibly soft, sticky, and profoundly sweet.
What do they actually taste like? For those of us who have never had the privilege of eating properly pit-roasted camas.
According to historical accounts from early settlers and from modern indigenous groups who are actively reviving these traditional foodways, the flavor is incredibly complex and rich. They are most frequently compared to the taste of heavily roasted pears, baked sweet potatoes drenched in syrup, or pure molasses.
That sounds delicious.
Long before European processed sugar was introduced to the North American continent, roasted camas was one of the primary sweetening agents utilized by native peoples. They would use the sweet black bulbs to flavor savory meat stews, or they would take the sticky cooked bulbs, pound them into a thick paste, form them into large flat cakes, and dry them in the hot summer sun.
A prehistoric energy bar.
Exactly. And because the moisture was removed and the sugar content was so extraordinarily high, these dried camas cakes acted as their own preservative. They could be stored safely for years without spoiling. They became a massive foundational trade commodity across the Pacific Northwest, second in economic importance only to dried salmon.
The nutritional yield from this engineering process is just staggering to consider. By combining the thermal mass of rocks, the specific acidic properties of skunk cabbage, and the physics of a sealed earthen pressure vessel, they achieved a 250% increase in digestible, bioavailable calories compared to the raw bulb.
They took a gastrointestinal nightmare and transmuted it into a life-sustaining, energy-dense staple.
But the engineering required precise execution. If the oven failed to reach the required temperatures, or if it lost its seal and the steam escaped, the hydrolysis wouldn't complete.
Right, which brings up a fascinating practical problem. When you bury your food under 3 feet of dirt for 3 days, you can't exactly open the oven door to peek inside.
And you certainly can't stab the bulbs with a digital meat thermometer.
Exactly. So how did the indigenous cooks know, empirically, if the exact thermodynamic and chemical thresholds had been met?
I read about this, and it is amazing.
They utilized human biology as the ultimate indicator. Specifically, they used flatulence as a biological metric.
That is just brilliant. Explain how that worked.
It was an entirely empirical process. When a community opened a massive earth oven, the families would eat a portion of the camas. If, later that evening or the next day, the people who ate the camas began experiencing severe gastrointestinal gas, the experienced cooks knew instantly and definitively what had happened.
The presence of gas was empirical, biological proof of incomplete inulin hydrolysis.
It meant the oven had failed. The rocks hadn't been heated long enough, the pit had a leak, or the gathered vegetation wasn't acidic enough.
That is the most rigorous scientific application of a fart joke I have ever encountered. Listen, we've got gas rolling through the camp. The internal pH of the reactor clearly didn't drop below 4.
It is a perfectly calibrated biological feedback loop.
But taking a step back, when you realize that safely acquiring and digesting this plant requires navigating a lethal neurotoxic minefield, hand-pulling poisonous mimics, and engineering an underground acid hydrolysis reactor, it fundamentally changes how you view the people and the place.
It completely shatters a deeply ingrained modern misconception.
Right. Surviving the poison and mastering the complex chemistry implies a level of interaction with the landscape that goes vastly beyond the casual concept of foraging. You do not just stumble upon a 72-hour underground acid catalyst process while taking a walk in the woods.
No, you don't.
This leads us to a fascinating realization about the landscape itself. These wet prairies weren't just wild, chaotic fields. They were heavily managed, meticulously engineered agricultural zones.
This touches on what anthropologists and ecologists call the romantic myth of the pristine wilderness. For centuries, European settlers and modern history books perpetuated the idea that before colonial contact, the Americas were just vast, untouched, wild expanses where native people simply wandered around, passively picking whatever happened to grow out of the dirt.
The concept of the noble savage living lightly on an unmanaged earth.
It's a pervasive myth, and the ecology of the Camas Prairie proves that it is entirely false. The wet prairies of the Pacific Northwest were essentially massive, multi-generational gardens. They were complex agricultural landscapes terraformed over millennia by tribal nations like the Coast Salish, the Kalapuya, the Samish, and many others.
To understand the sheer scale of this terraforming, we have to look at their macro-ecological management strategies. You found a term in the literature during our research that perfectly encapsulates this: the Coast Salish Three Sisters.
Yes, the Three Sisters. Now, if you grew up in North America, you likely learned about the Mesoamerican or East Coast iteration of the Three Sisters, corn, beans, and squash, growing together in a single symbiotic mound of dirt.
Right. The corn provides a stalk for the beans to climb, the beans fix nitrogen in the soil, and the broad leaves of the squash shade the ground to retain moisture. It's a beautiful microagricultural system.
But the Coast Salish version of the Three Sisters operates on a totally different scale. It is a much broader, ecosystem-wide companion planting concept. Instead of 3 plants in a single mound, it refers to the macro management of 3 vital, interconnected food staples that define the entire regional ecology: the Gary oak tree, the blue camas flower, and the Pacific salmon.
And the primary agricultural tool they wielded to manage all 3 of these wildly different resources simultaneously was fire.
Fire, they were intentionally setting the prairies ablaze.
Yes, indigenous land managers used highly controlled, low-intensity burns to actively sculpt the landscape. Let's look at the Gary oak and the Camas first. Both of these plants are sun-loving species. They require open, sun-drenched savannas to thrive.
But the natural ecological trajectory of the Pacific Northwest is to become a dense, dark, impenetrable coniferous forest.
Trees like the Douglas fir grow incredibly fast in that climate. If you leave a wet prairie alone, within a mere 30 or 40 years, it'll be completely choked out by Douglas fir saplings.
And those fir trees cast a deep, permanent shade that eventually kills off the camas and the Gary oaks.
Now, the indigenous people used fire as a massive landscape-scale weeding tool to hold back the forest.
Precisely. They would meticulously burn the prairies on a regular, calculated cycle. Modern ecological research, specifically studies conducted on Waldron Island in the San Juan Archipelago, analyzed historical fire scars on ancient trees. The data show that prior to the 1880s, these exact prairie areas were intentionally burned on an average cycle of approximately 7.4 years.
Every 7 years, a cleansing fire sweeps through.
This specific low-intensity fire would creep through the grass, killing the young, thin-barked Douglas fir saplings and burning up the dead brush. But the mature Gary oak trees possess thick, corky, fire-adapted bark, so they survive easily.
And the Camas bulbs.
They're buried safely several inches underground, so they are entirely protected from the radiant heat passing above them. In fact, the fire was extremely beneficial to the Camas. It cleared the dead thatch, opened the soil to the sun, and the ash returned vital concentrated nutrients like potassium and phosphorus back into the earth, priming the soil for a massive Camas bloom the following spring.
And maintaining this 7.4-year cycle has a massive secondary benefit that is incredibly relevant today. It completely prevents the catastrophic, crown-destroying wildfires that are currently devastating the American West.
By burning frequently, the indigenous managers constantly eliminated the latter fuels.
The buildup of dead brush, fallen branches, and dense undergrowth that allows a small ground fire to climb up into the forest canopy and become an uncontrollable inferno.
The historical contrast is stark and really tragic. Once colonial land management policies took over in the late 19th century, they aggressively outlawed all indigenous burning practices under the misguided belief that all fire is bad for forests.
And the result?
According to the research, the fire interval in those exact same San Juan Island areas jumped from a managed 7.4 years to a staggering 103.8 years. We forcibly removed the gardener from the garden, the underbrush piled up into a massive tinderbox, the Douglas firs aggressively encroached and shadowed out the savannas, and as a direct result, we lost 91% of the native prairies in the Pacific Northwest.
91%. Today, only about 3% of those historic vibrant prairies remain productive with native species. It is an absolute ecological tragedy born of ignorance.
It truly is.
But I want to connect the final dot and the Coast Salish three sisters. You have the elk and the Camas thriving in the fire-cleared prairie. How does burning a terrestrial field of grass help the third sister, the Pacific salmon? A fish lives its entire life in the water. How does a fire on land benefit a marine animal?
This is where the profound, interconnected ecological reciprocity of indigenous science truly shines. When you burn thousands of acres of coastal prairies, two vital things happen that directly impact the aquatic ecosystems. First, the massive amounts of ash generated by the fires eventually wash down through the watershed and into the nearshore aquatic habitats and estuaries.
The ash acts as a fertilizer.
Exactly. This ash provides a massive sudden nutrient boost of carbon and minerals to the coastal eelgrass beds. Eelgrass meadows are the primary critical nursery habitat for juvenile salmon preparing to enter the ocean. By burning the land, they were fertilizing the ocean nurseries.
That is incredible. And the second benefit?
Temperature control. The late summer and early fall is when the adult salmon leave the cooling depths of the ocean and begin their arduous journey up the rivers to spawn. If the river water is too hot, the salmon become stressed, diseased, or die before reproducing. During the heat of the summer, the vast, lingering smoke from these controlled prairie burns would actually pool in the valleys and provide a thick, cooling canopy cover over the rivers and coastal waters. This smoke layer blocked the intense summer sun, physically lowering the water temperatures to levels that were significantly more hospitable for the returning salmon runs.
Let's just pause and appreciate the staggering scale of that environmental understanding. By tending their terrestrial root gardens with fire, they were simultaneously fertilizing the marine nurseries with ash and literally air conditioning the spawning rivers with smoke.
They were terraforming across biological kingdoms, and this profound understanding wasn't just practical, it was deeply embedded in their cultural and spiritual frameworks. There is a well-known Coast Salish oral history about Salmon Boy, a human child who is taken to live with the salmon people deep in the ocean. He learns their ways, understands what they need to survive, and eventually returns to the surface to teach human beings how to properly steward the waters and the land to ensure the salmon always return.
These stories weren't just whimsical fairy tales. They were the encoded, generational, scientific operating manuals for managing an entire biome.
Exactly.
So they're managing the macro ecosystem with fire and smoke. But let's bring it back down to the micro level, back to the dirt, and back to our lethal twin, the death camas.
Right, the zygosine hazard.
If they're cultivating these massive, sprawling fields of blue camas to feed thousands of people, how are they ensuring they don't accidentally harvest the death camas? You can't use fire to kill it because the death camas bulb is buried safely underground right next to the blue camas bulb. The fire passes right over both of them.
You are absolutely correct. Fire is a blunt instrument that cannot differentiate between the toxic and the edible. Removing the death camas required meticulous, dangerous, and incredibly tedious manual labor. It required generations of visual discipline. And this brings us back to that narrow window of clarity we discussed at the very beginning of the deep dive, the brief spring blooming period.
Right, that short window from late April to mid-May, when the blue flowers of the edible camas and the creamy white flowers of the death camas are both visibly blooming above the grass.
During that specific fleeting time, indigenous land managers and historically, this vital life-saving work was almost exclusively the deeply respected domain of the women in the tribes would systematically walk through their designated family prairie plots. They didn't just casually stroll through, they grid-searched the land.
They're looking for the white flowers.
They would visually identify every single death camas plant by spotting its creamy white flowers and its yellow-green nectary glands, and they would manually dig them out of the soil. They quite literally weeded the death out of the garden.
Think about the dedication that requires. They walk through hundreds, maybe thousands of acres, pulling a lethal neurotoxin out of the ground one bulb at a time, and they had to do it perfectly.
Perfection was the only acceptable standard. They would extract the toxic bulbs, stems, and flowers and carefully dispose of them away from the food plots, ensuring the soil was purified. By the time the spring flowers died back, the stalks withered, and the actual late summer harvest season began when all the plants looked like identical dry grass they knew, with absolute empirical certainty that every single dormant bulb remaining under the soil in that specific managed plot was a safe, edible blue camas.
But nature is chaotic. Nature makes mistakes. What happens if a blue camas plant experiences a random genetic mutation? So I read in our research that occasionally a perfectly safe, edible Camas will produce a genetic mutant that blooms with white flowers instead of its signature blue.
That is the ultimate botanical curveball. You have a perfectly safe, edible plant that is packed with nutritious inulin, but because of a random recessive gene, it throws up a white flower, making it look almost exactly like the toxic death camas from a distance.
If I were managing that field and I knew how much effort it took to grow a Camas bulb, I might be tempted to walk up to that white flower, look really closely to see if it lacked the yellow-green nectary glands, and if it was just a mutant blue Camas, leave it in the ground for harvest.
And that temptation is exactly what their cultural protocols were designed to eliminate. The rule governing the harvest was absolute and uncompromising. If it blooms white, it goes.
No exceptions.
None. Strict, community-enforced cultural protocols dictated that all white flowering bulbs, without exception, be immediately destroyed. They did not take the risk of closely examining a white mutant to see if it lacked the venomous glands. They didn't trust a visual inspection when the stakes were the SLUDG, toxidrome, and respiratory failure.
To completely eliminate even the microscopic risk of mistaking a mutant edible plant for a lethal toxic one, they simply eradicated any white bloom from the food plots.
It is the ultimate expression of better safe than sorry. You simply do not gamble with zygosine. It paints such a vivid picture of this intense, ongoing, respectful relationship with the land. The prairie wasn't a wild, chaotic grocery store where you hope to get lucky. It was a highly curated generational farm that demanded constant vigilance.
And their agricultural stewardship extended to the physical structure of the soil itself, didn't it?
It did. When the late summer harvest finally arrived, they didn't use broad plows or massive shovels to rip up the earth. They used highly specialized, narrow, T-shaped digging sticks, traditionally carved from dense hardwoods, like ocean spray, or fashioned from elk antler.
Because a shovel would just destroy the soil.
Exactly. Instead of violently churning the earth and destroying the delicate fungal networks in soil structure, these narrow sticks allowed the harvesters to precisely target and selectively pop only the large, mature camas bulbs out of the ground.
It's surgical harvesting.
And while they were levering these mature bulbs out, the plunging action of the sticks was actively aerating the dense prairie soil, creating little pockets that captured fall rainwater. Furthermore, when they dug up a large mature bulb, it was usually surrounded by a cluster of tiny, immature bulblets.
What do they do with those?
The harvesters would carefully break off these small bulblets and immediately replant them right back into the freshly aerated holes. They were actively expanding and propagating the camas populations, ensuring that the crop would be even denser for the next generation.
So as we draw this massive journey to a close, what does all of this mean for us today? As we look back on everything we've unpacked in this deep dive, the terrifying mimic morphology of the bulbs, the venomous nectar feeding the solitary miner bee, the complex indigestible inulin chains, the brilliant thermodynamic engineering of the acid catalyst earth ovens, and the sweeping landscape-scale fire ecology of the Three Sisters, it fundamentally shifts your perspective on human history and intelligence.
If we connect all these individual threads into the broader tapestry of human evolution, it forces a profound, humbling respect for the depth of ancestral science. Safely eating a camas bulb wasn't a matter of luck. It wasn't the result of a primitive hunter-gatherer stumbling through the woods and taking a bite out of a root. It was the culmination of thousands of years of rigorous, empirical stewardship.
It's staggering to think about the generations of trial and error that must have gone into perfecting this system. How many early ancestors suffered the terrifying paralysis of the SLDG toxidrome from a misidentified bulb before the strict protocols of the spring weeding were established?
Or how many communities suffered through the agonizing balloon factory fermentation of undercooked inulin before a brilliant, forgotten indigenous chemist realized that adding acidic skunk cabbage and sealing it underground for exactly 3 days was the magical thermodynamic key.
It required safety engineering to systematically remove the death camas, advanced thermodynamic mastery to construct the subterranean ovens, intuitive chemical knowledge to utilize the volatile acids from surrounding foliage, and a deep, reciprocal, multi-generational relationship with the entire ecosystem to maintain the prairies with fire.
It is a testament to human ingenuity and the intimate, necessary relationship between a people and their environment. The knowledge isn't just deep, it is breathtaking in its precision.
Which leaves you, the listener, with a final, provocative thought to mull over as you go about your day. We look at a plant like the blue camas, a plant that maliciously stores its energy in a carbohydrate matrix our mammalian bodies literally cannot digest, and that chooses to grow side by side with a neurotoxic mimic that will stop your heart. To the untrained modern eye, that looks like a completely useless, highly dangerous, hostile landscape.
We would look at it and pave over it.
But through generations of patient observation, brilliant chemical engineering, and ecological stewardship, it was transformed into the sweet, sustaining foundational cornerstone of an entire civilization's diet. So it makes you wonder, as you look around our modern world, what other things do we currently dismiss as useless or fear as dangerous elements in our environment? Are they truly useless, or are they actually profound gifts just waiting for the right shift in perspective or the rediscovery of an ancestral technology to finally unlock their potential?
That is a vital question for how we choose to view and interact with the natural world moving forward. There is always a deeper layer to uncover if we are willing to look.
Thank you so much for joining us on this incredibly deep dive into the wet prairies. Keep exploring, stay curious, respect the chemistry of the wild, and we will catch you on the next deep dive.